So I'll talk about one particular subject that actually leads into another subject today. But the lab is working on a lot of things, and I sort of enjoy working on a lot of things. And some of that has to do with the corals and the symbiotic association between the alga, which is a dinoflagellate, and its animal host. This has to do with photosynthesis and a collaboration with people at mechanical engineering, where we're actually looking at the thylakoid membranes and the complexes on the thylakoid membranes. For example, this is a PS2 dimer and the oxygen evolving complex associated with PS2 in the membranes, and trying to develop that system to look at dynamics of those membranes in real time when you shine high light on the membranes, when you change the redox conditions, when you inhibit the photosynthetic apparatus. We also do a bit of work on the desert crusts, and that's something I'm more interested in developing. There are many organisms in those crusts. Most of them are prokaryotic, but there are some eukaryotic algae. And those algae, when they've been isolated, they have incredible characteristics. For example, they're the fastest growing eukaryotic algae that I know of. And the doubling time could be about 2 hours under optimal conditions. And they also can take incredibly high light. You could put them at 2,000 micromole per meter squared per second photons, and they're not photoinhibited. And you could even raise that to 3,000, and they just do a little more photosynthesis. So most organisms would be dead at those light intensities, but these organisms survive. We also do a lot of work on Chlamydomonas, and I'll tell you a little about the green cut today. That's a fascinating project to me, and it developed out of the genome project. We started the genome project here, and once we got the Chlamydomonas genome, I really didn't want to report on another genome. I wanted to use it in a comparative way. And the green cut and the cilia cut gave us more information about functionality. And we were able to use that genome very effectively to learn about functionality. We also work on anoxia and do a little work with companies, in fact, to make oils from algae. And I'm also very interested in what I call the energetic landscape or network of electron transport. And can anybody actually identify this?
New York.
You got it. It's the New York subway system. And that's what I think about when I think about these electron flows. They're incredibly complex. And if you think about it, you know, light is intercepted by a plant. It's absorbed by chlorophyll. That generates a charge separation and electron flow. And in fact, that light energy is really converted to electrical energy. And it's all these circuits that are generated, ultimately, that give you chemical bond energy, and it could also be quenched very effectively. So how that really works and these interactions are interesting for some of the future work that I'm planning. Now today, I'll talk about a particular project which has to do with the biogenesis and evolution of an organelle. And that will lead into how we get to specific functions of new or novel proteins involved in photosynthesis, how we know they're involved in photosynthesis, and how we begin to probe those. Okay. Now, there is a reason I actually took this project and developed it for this talk, and it's because I thought it reflected the Carnegie tradition. A tradition that I came into when Winslow actually hired me, and that was in 1982. That was a long time ago. I guess time is all relative. So. But the idea was develop new projects and concepts. Look for areas that have not really been developed before, but that you think are important. Don't chase money. In fact, that's what a lot of us do these days. We really chase money. And there's a good reason for that. You can't do the work without money. and a lot of the agencies are dictating a bit what you do. But there was a time at Carnegie when, in fact, you weren't allowed to chase money. You couldn't get federal grants. It was not looked at kindly. So, allow postdocs to take projects with them. This could be, in fact, some of the most influential influence that you have on people by letting them develop into independent researchers and starting with a project that they actually initiate at Carnegie. Try not to do what can be done in a university. We're not a university. We have flexibility. We have some hard money. So we could actually explore things that sometimes a university has more constraints, financial constraints and other constraints. And I think this is sort of typified by the work of Eva Novak, who worked on a little-known organism called Polynella chromatophora. And she was interested in the evolution of organelles, how organelles actually evolved. And this is an interesting organism to work with for a number of reasons. Now, Eva contacted me, and she said, I have this organism, and this is what we're doing, and this is some of the work we've done on it. But I can't do the biogenesis where I am. I can't do this, and I want really to collaborate on this project, and I'd like to come to Carnegie. So we wrote a grant together, and she got a grant to come to Carnegie. And it really was a collaboration. Now let's start off with the endosymbiotic origin of eukaryotic plastids. Most of you know that the plastid, the chloroplast, started its evolution about 1 billion years ago when a protist engulfed a unicellular cyanobacteria, and that diverged into 3 groups, one group into the green lineage, land plants and green algae, another into the glaucophytes, and another into the red algae. People think, or I guess the dominant thought is that this is a monophyletic event. There are some people who do not think it's a monophyletic event and that it happened more than once. But it's a very hard event to happen and stabilize. And there are a lot of reasons for that, which we'll talk about a little bit. Now, when this type of thing happens, there's an interaction between symbiotic partners. The 2 partners have to come together, and there has to be something that locks them in place. Often these protists will just engulf a bacteria and digest it. And it probably digested a lot of cyanobacteria before the event became fixed. Once the event becomes fixed, there's gene exchange from the engulfed cyanobacteria to the nuclear genome of the host, and that's called endosymbiotic gene transfer. There's protein exchange going on, there's metabolic exchange going on, and there has to be integration of metabolisms. And if you really think about it, when a cyanobacteria absorbs light energy, you're exciting pigment molecules. You're driving electron transport. You're generating ATP and NADPH. But if there's not a good connection between the endosymbiote and the host, the endosymbiont can't grow much in the host, and there can't be the exchange of those metabolites initially. And you'll back up the whole electron transport system. You'll generate excited electrons, which give you reactive oxygen, and you'll basically die. You'll burn up the cell. So this integration ultimately is important. And you might think at the initial stages of forming this type of association that the organism would be light sensitive, high light sensitive. And in fact, Polynella is light sensitive. I wouldn't say it's high light sensitive, it's pretty low light sensitive, right? You don't have to go very high to damage this organism. Now, this association, Polynella, it's between a protist, and this is a thecate protist, and that's shown here, and a cyanobacteria. This cyanobacteria was engulfed here, and that cyanobacteria in this organism is called a chromatophore. It's not an independent organism anymore. It can't be grown outside of the host. So it's really an organelle of some type. This engulfment occurred between 60 and 200 million years ago. It's not really clear exactly when it occurred. The organism itself is in the sediments in freshwater ponds and lakes. It's in the sediments because it is high light sensitive. So it found a niche where it can live. Its phylogenetic affiliation is the Rhizaria. It's a Cercosian Rhizaria. It's vertically transmitted. That is, the organelle is transmitted from one generation to the next. And you could see it's thecate. It's a large organelle. In fact, when I look at this, this is a huge cyanobacteria. If that was its, or if it was like that in nature, it probably wasn't. That actually looks like a cyanobacteria that's stressed. When you stress a cyanobacteria, it begins to elongate. So, I don't know, Synechococcus, it might be 4 or 5 microns. And when you stress it, it filaments a bit, and it could become 20 microns. And in fact, this organelle, there are 2 of them in here, are about 20 microns when you take them out. This is the closest relative. It's called Polynella ovalis, and it doesn't have the organelle. And so you can actually compare the heterotrophic organism with the now photosynthetic organism. And if you look at the ultrastructure of the cyanobacteria in here, it looks like a typical cyanobacteria. You could see the thylakoid membranes, you could see the carboxysome, you could see the peptidyl glycan cell wall, which is still around it. It's a bit ephemeral at this stage. Now, if you look at the DNA in the organelle, so if you look at DNA in the progenitor, the presumed progenitor cyanobacteria, it's between 2 to 5 megabases. If you look at the DNA in a chloroplast, it's usually between 0.1 and 0.2 megabases. But if you isolate the DNA from the chromatophore, you find it's 1 megabase. So it's right in between. It's an organelle that's in the process of reducing its DNA. Okay. Now, what's on that DNA? What does that DNA encode, and what does it really tell us? Well, it encodes many biosynthetic pathways, and many biosynthetic pathways have either been lost or partially lost. That is, particular genes that are required for that biosynthetic pathway are no longer on the chromatophore genome. And if we look at one biosynthetic pathway, here's the pathway to make chlorophyll and phycobilins. And the gene that's been lost on the chromatophore genome is heme-D. Now, if you lost this gene, you could imagine a couple of things happening. For example, you can go up to heme-C in the chromatophore, then that product could go outside into the host cytoplasm, where heme-D converts it to uroporphyrinogen 3, and then you would have to come back into the chromatophore and continue the pathway. Or the protein for heme-D, which is now made in the cytoplasm, would have to come into the organelle and fill that pathway and basically serve to patch that pathway by an import process. And so there has to be metabolic trafficking, protein trafficking, displacement of function from the chromatophore to the nucleus. And that displacement of function from the chromatophore to the nucleus, and so this was displaced from the chromatophore to the nucleus, is often endosymbiotic gene transfer, or it could result from horizontal gene transfer. That is, genes from outside the organism could come in and replace functions too. Or the gene in the nucleus of the amoeba might also replace functions if it has an analogous gene that could do that function. So there are a lot of possibilities here. But the interesting thing to me about the chromatophore, if you look, all the transporters for nitrogen are gone. So the chromatophore genome no longer makes the transporters for nitrogen. So those transporters have to come from the nuclear genome. because the chromatophore has to take up nitrogen compounds. So the host has to provide either metabolites or proteins, transporters here. Now, photosynthesis is going on in the chromatophore, and the chromatophore is providing carbon to the host. So there's an exchange. And here you could see these transporters have to come in, have to be somehow provided by the host. And those are host-derived transport systems. Now, we could look at some of these genes that are actually have been transported from the genome of the chromatophore to the host and asked what happens to those proteins. Do those proteins actually get into the organelle and fulfill a function in the organelle? And if you look here, there are over 30 nuclear genes of alpha cyanobacteria origin in the host nuclear genome. So these are presumably endosymbiotic gene transfers. They came from the chromatophore genome, they were transferred into the host genome. There's a bias toward short genes, and a number of them function in photosynthesis and light acclimation. And some of the genes, PSAE, PSAK, are EGT genes, endosymbiotic gene transfer genes. There's 2 copies of PSAE, 4 copies of PSAK on the host nuclear genome. And here you could see that PSAE and PSAK are part of photosystem 1, and they're required for optimal functioning of photosystem 1. So you would imagine that photosystem 1 has to have those proteins. So the questions really are nuclear encoded photosystem 1 subunits synthesized in the cell's cytoplasm. They're encoded on the nuclear genome. Do they get to their site of function in the chromatophore? And if they do, can they assemble into PS1? And how do they get there? Okay, so those are the questions we posed initially. And here is a sort of repeat of that. Are EGT genes transcribed in the nucleus and translated in the cytoplasm on ADS ribosomes? If they are, do they enter the chromatophore? If they get into the chromatophore, do they associate with the appropriate complex? How do they get into the chromatophore? There's no TIC-TOC system, which is the protein transport system that takes proteins from the cytoplasm in plants into the chloroplasts. Okay? So it has to be a different system by which they get into the chromatophore. Okay, so we did a very simple experiment initially, and that is we did immunogold localization of PSAE. That's encoded on the host nuclear genome. And is it in the chromatophore? And if you look at the labeling, this is pre-immune serum, and you see very little labeling. This is the chromatophore. This is the cytoplasm of the host. Now, here's labeling with PSAE antibody. And you can see the immunogold particles are over the chromatophore and not over the host region of the cell. And you can see it's pretty densely labeled over the thylakoid membranes. So it looks like this PS1 protein is getting into the chromatophore, and you could see it on the thylakoid membranes. And here is a blow-up of that, and you can see many of the gold particles here. Okay, so that told us it's in the thylakoid membranes of the chromatophore. Now, are PSAE and PSAK, which is another EGT protein, assembled with PS1 in the chromatophore? In order to show that whether they're assembled with PS1, we had to isolate PS1 from the chromatophore. So it was good old-fashioned biochemistry that we used, and it took a little while, but ultimately Eva got very good PS1 complexes out of the chromatophore, and PSAE is here, PSAK is here. We had antibodies, and this shows the antibodies against PSAE, that this is PSAE. And we also sequenced these proteins out of the gel using mass spec. And you can see the protein sequences here. So in fact, we were able to detect 2 PSAKs in this band here, 1 PSAE here, but there are actually 2 and 4 of these proteins that are encoded on the nuclear genome. Now, usually when you look at a protein that gets into an organelle, it has a pre-sequence. It has a tag on it that gets it into that organelle. When we looked in front of the N-terminus of these using genomic sequence, we really saw no pre-sequence on these proteins. So we concluded is that they must get into this organelle without that tag. So some low molecular weight PS1 subunits are nuclear encoded. They enter the chromatophore and they assemble into PS1 complexes. They likely enter the chromatophore without a pre-sequence. Okay. Now, another classical experiment to really confirm this type of thing, you could imagine maybe this subunit, maybe the RNA gets into the chromatophore. I think that's very unlikely. But if you want to make sure that that's not the case, you could see whether the protein synthesis, whether these proteins, their synthesis is inhibited in the presence of chloramphenicol or cyclohexamide. Now, chloramphenicol inhibits translation on 7DS ribosomes in the chromatophore or in cyanobacteria, while cyclohexamide inhibits translation on 80S ribosomes in the cytoplasm of the cell. And if we look at this, in the presence of chloramphenicol, we block a lot of proteins associated with PS1. So this is PS1 particles, and we labeled proteins as they were synthesized, and we did it in the presence of cyclohexamide, in the presence of chloramphenicol, in the presence of cyclohexamide, or both. Here, PSA-A and PSA-B are inhibited in the presence of chloramphenicol. They are inhibited a bit in the presence of cyclohexamide, but you would expect that because if you block the biogenesis of the complex, the other proteins, even if they're not their synthesis is not inhibited, they won't get into that complex that well. But what you can see here clearly is that many of the proteins are inhibited by chloramphenicol. In fact, all of the proteins that are encoded on the chromatophore are inhibited by chloramphenicol, whereas PSAE is made in the presence of chloramphenicol. It's lighter, as you see, and PSAK is made in the presence of chloramphenicol, but it's blocked when cyclohexamide is used, which really says that the RNAs for these proteins are translated in the cytoplasm and that the proteins must get into the chromatophore. Okay. So PSAE and PSAK are both synthesized in the cytoplasm of this cell. Now, the other thing that we noticed was very interesting. When we did the immunogold, of course, we saw the gold particles this is for PSAE, were concentrated over the thylakoid membranes, which we expected. But we also saw gold particles concentrated over the Golgi. And that's actually shown here. This is the Golgi here, and you can sort of see the gold particles here. This is a blow-up. And the gold particles are concentrated over the Golgi, not quite as densely as over the thylakoids. And you can see if you quantify it, this is the PSAE over the chromatophore. This is pre-immune serum. This is over the Golgi. You could see they're concentrated quite a bit over the Golgi. So it suggests that PSAE may be routed to the chromatophore via the secretory system. And maybe this was an early promiscuous way to get proteins into a developing organelle. So what did we learn now about protein transport and into this organelle? What we know is here. We know that these EGT proteins are synthesized on 80S ribosomes. So they're synthesized in the cytoplasm. We don't know that they get into the ER, but we assume that they're getting into the ER because it looks like they end up in the Golgi. And that we know because we could see that with immunogold. Now, from the Golgi, they have to somehow pass through the double membrane of the chromatophore through the peptidoglycan cell wall and end up in the thylakoid membranes in PS1, which we do know. But this area of transport here is a black box at this point. But it is telling us, I think, about the early evolution of communication between the cytoplasm and a nascent organelle. So chromatophores evolved independently of plant and algal plastids. They have a highly reduced genome. Many pathways are lost or incomplete. There's highly reduced transport potential encoded on the chromatophore genome. For example, there are no nitrogen transporters. There's at least 30 cases, there's more than that, of EGT, endosymbiotic gene transfer. Most transfer genes encode small proteins that might be easily transferred into the organelle. Many of these proteins have to do with light in some way or another. The nuclear encoded PS1 subunits, they're synthesized on ADS ribosomes, localized to chromatophores, assemble in PS1, and the Golgi might function as an intermediate in trafficking into the chromatophore. EGT in combination with re-import of proteins is defining really the features of a canonical organelle. So the chromatophore, like a chloroplast, is a canonical organelle. But you have to think about this. Integration may still be very suboptimal. It's extremely light sensitive. Transport of metabolites between the compartments may be slow and inefficient. And the organism is in the process of searching for an answer to this problem of integration in evolutionary time. Now, how does Paulinella respond to light? And is there light regulation of any of these EGT or photosynthesis genes? And when we look at cyanobacteria, many of these genes are actually controlled by light. And they're upregulated in light, like the PS1 genes, and often it's blue light that's controlling them. Are they controlled by light in this organism? And the person who did a lot of this work on light regulation is Ru Zhang, and she's currently a staff scientist at the Danforth Center. So let's look at sort of the coding capacity in Paulinella. Most of the photosynthetic genes are encoded on the chromatophore. So they're encoded on the chromatophore, the proteins are made in the chromatophore, and assembly occurs in the chromatophore. A number of the photosynthetic genes are EGT genes. We talked about PSAE and PSAK, but there's also a PS2 gene, PSBN, and there's also a family of genes called highlight inducible genes, which I'll talk about a little bit. Now, there's also some genes that are encoded on both the chromatophore genome and the nuclear genome. And that's a PS1 gene, PSAA, and a carboxysome gene. PSAI and a carboxysome gene. Okay, now if you think about that, if you have a gene on both genomes, it's probably in the process of evolving. It'll lose one probably or the other. One will become more efficient, one will become more selective, and will be selected over the other. Okay, so you can actually see evolution in process in these genes. Now, if we look at growth of these organisms in the light, this is 15 micromole photons, 30, 60, 90, 120. You could see by 90, they're really suffering, and they're actually dying. By 120 micromoles, this is after 3 weeks, they're dead. Okay, it takes a little time for them to die. They try to acclimate initially, but they can't make it. And so they're very sensitive to light, and of course they found a niche to live in, which is sludges on the bottom of ponds, so they never see that much light. Okay, now are any of these genes controlled by light at all? Now this shows you the photosynthetic genes that are the EGT genes, so they're in the nuclear genome, and you could see PSA, PSBN, PSAE, PSAK, all the different subunits of PSAK. And you could see there's not much light control at all. So they're not really controlled by light in this organism. But there's another set of EGT genes which are called the HLIs. And actually, we found the HLIs back in the 90s. And we studied the HLIs, and we found a number of things about them. They encode single membrane helix proteins that resemble the first and the third helix of the LHC proteins. They represent the progenitors, probably, of the LHCs. In cyanobacteria, we made mutants in these genes, and they became incredibly photosensitive. And so the proteins, the HLI proteins, are somehow involved in managing light energy and allowing the cells to survive in the presence of light. Now, they may be involved in quenching. They may be involved in chlorophyll biosynthesis to some extent. They may interact with intermediates in chlorophyll biosynthesis or free chlorophyll, because those intermediates are very toxic when they bind light, when they absorb light. And so they may be bound, and in the case when they're bound, they're not really toxic. They also may be involved in repair of the photosynthetic apparatus. Now, in cyanobacteria, there are usually about 4 to 8 genes for the HLIs. In highlight ecotypes, like the highlight ecotype of Prochlorococcus, I think there are either 21 or 24 of these genes. So in organisms that are exposed and grow in habitats that, where the habitat has a highlight component, this gene family seems to have expanded. In Paulinella, there are over 30 nuclear encoded HLI genes. So it's as if the gene moved to the nuclear genome from the cyanobacterial endosymbiont, and the gene it duplicated and it expanded into a large gene family. And actually, if we look at some of these genes, they're controlled by light, which actually amazes me. And so here you can see these are 3 different HLI genes, and this is light given for a certain amount of time at these different intensities. And you could see they're upregulated by light. Okay, so there are many of these HLIs are controlled by light. So when you think about it, they had to get into the host genome, they had to get promoters and terminators and regulatory sequences that allow them to be controlled or activated in light. And what type of light are they controlled by? Well, this is one particular HLI gene, and in white light you could see the level of expression, the level of mRNA goes up pretty high in white light, but at very low levels of blue light it's upregulated very high. So they're blue light regulated genes. And of course, there are blue light photoreceptors in both the chromatophore and in the host. And so somehow these probably have gotten into the host genome and under the control of promoters that are controlled by blue light photoreceptors. Now, this is something that we grappled with. When we think about the evolution of an organelle, should we really just be thinking about endosymbiotic gene transfer and the genes that are already in the genome of the protist with respect to that evolution? How much could horizontal gene transfer possibly shape the activity of the developing plastid? And this is an area that I was a little surprised in terms of the results of this, and I'll show you some of those results. Let's look at some of these pathways again. So this represents the chromatophore, which is delineated by a green line here, and this represents the cytoplasm. Now, the pathway for peptidoglycan biosynthesis is mostly encoded on the chromatophore genome. It's missing one gene, MURF. MURF is found in the nuclear genome. So that gene has migrated to the nuclear genome. This is still making a peptidoglycan cell wall, so the pathway still has to be active. And if you look here, this is MURE, so this is the product that would be worked on by MURF to make this product. You could imagine that this particular product This may go outside the cell, be processed by μF, the product then come back in, and you complete the pathway. Or the μF protein, just like the photosystem 1 proteins, go into the chromatophore and fill that gap. Because these are big products, it's hard to imagine that they do get out and come back in, but that still is possible. You could look at some other incomplete pathways, like for DNA replication. Here you're missing 2 components of the DNA replication apparatus on the chromatophore genome. Those are the genes for Lig-A, the ligase, RNase ligase, and RNase H, or POL1. Those are encoded on the nuclear genome of the host. Now, you can't have a product going in and out for this, so the proteins must get in and fill these gaps. So now we know that in almost all cases that we looked at, the proteins appear to come in from outside and fill any losses that occur on the genome of the chromatophore. Now, what are those proteins? What are their origins? You might expect the origin to be alpha cyanobacteria, which is what the chromatophore is. You might expect them to be generated through endosymbiotic gene transfer, but most of them are not. And if you look here, most of the proteins that fill these gaps are from a whole range of other bacteria. And you could see the different phyla here. I think there are 7 or 8 phyla here that they represent. So horizontal gene transfer is one of the major processes that allows the reduction of the chromatophore or plastid genome. It's compensating for losses on genes on that chromatophore genome. Now, at first you might think this is a little bizarre and how does this really happen? But then you think about the history of these organisms. And initially, Paulinella was a heterotroph. It didn't have a chromatophore. And so what did it do? It ate. And what did it eat? It ate bacteria. And so here you can see it's ingesting bacteria. It's putting them in phagotrophic vesicles or vacuoles, and that's where it got its nourishment from. There were from some of these bacteria into the host genome, and so you'd get horizontal gene transfer occurring in this place, in this instance. Now, there would be a time when the organism was a mixotroph. That is, it could still eat and get genes through horizontal gene transfer, and it could also get genes through EGT, because now it's established an association with the cyanobacteria and is making it into a chromatophore. At this point, there would be a lot of HGT, horizontal gene transfer, and EGT. And in fact, this HGT could promote or support the loss of genes from that chromatophore. And that's what it looks like happens. And as you go on in the evolutionary process, you'll still have HGT, but now you have the eating. There's no eating. This organism no longer ingests bacteria. And most of the gene transfer would be through EGT. So that's how we think that's happening. Now, so I told you a bit about what we found out about the evolution of an organelle. Looking at an unusual organism that is in the process of reducing its the genome of its organelle, okay? And I've told you that you have EGT, you have HGT, and both are important in shaping that organelle. And I've also mentioned to you that it's almost an impossible situation when you have a photosynthetic organism in a non-photosynthetic organism, and you have to, over evolutionary time, tailor those interactions so you don't get a lot of reactive oxygen formed and so you don't kill that association. But what about photosynthetic function and the evolution of that? Now, to look at photosynthetic function, we use Chlamydomonas. We initiated a lot of the genome project here. And from that genome, we could look at gene structure. We could examine novel genes and genes families. A family that always interested me was the adenylyl and guanylyl cyclases, there are over 50 of these genes in Chlamydomonas. And we know what a couple of them do. A couple of them may be involved in mating of the organism, but we really don't know the function of most of those genes. We can use the genome to locate sites of insertion in mutant strains. We can perform targeted gene knockouts in some cases. We could find physical markers for map-based cloning of genes. We could identify a unigene set for various analysis to establish gene function, develop transcriptomics. But we could also do comparative genomics and deduce potential gene functions through those comparative genomics. And what maybe interested me most was using that comparative genomic analysis to look for genes involved in photosynthesis. And how do we really do that? Well, the project that I'll talk about has to do with the green cut, and that comparative genomic analysis is called the green cut. It's exploiting the informatic approaches to elucidate photosynthetic mechanisms. And there are lots of people involved in this project, including Shai, Tyler, Steve Herbert when he was here, and Wen Chung. But the work that I'll talk about today is mostly from Mark Heinekel and Rick Kim. Okay. Now, if you look at the Chlamydomonas genes and you see how well they match to Arabidopsis, or the proteins, how well they match to Arabidopsis proteins or human proteins, you could see some of those proteins match very well to human proteins. For example, this one here matches well to human proteins, but not to Arabidopsis proteins. While some of them, for example, this one here matches very well to Arabidopsis proteins, but not to human proteins. And that's because Chlamydomonas is half animal and half plant. And a lot of proteins in a lot of genes in Chlamydomonas that were lost in the streptophyte lineage during the evolution of plants were maintained in Chlamydomonas. And that's true, for example, for the cilia or flagella gene proteins. Here you can see these red ones are Chlamydomonas flagella proteins, the red and the pink. And they all match the human proteins very well. And of course, plants don't have cilia or flagella. Whereas the photosynthetic proteins, which are here in green, match the Arabidopsis proteins much better than the human proteins. But there are many, many unknowns that both match very well to Arabidopsis proteins or match very well to human proteins. And that's over half the genome. Now, can we predict new genes associated with chloroplasts or photosynthetic function? And how would we do that? And so we developed an informatic platform to actually do that. We created proteins based on the Chlamydomonas gene models. So there were about 15,000 or so proteins that we generated from those gene models. We then did a mutual two-way blast of each Chlamydomonas proteins with those of both photosynthetic organisms and non-photosynthetic organisms. In fact, in this, the photosynthetic organisms we used were the green lineage photosynthetic organisms, and they were plants and algae. And then we retained all proteins from all organisms that have a mutual best blast hit with the Chlamydomonas protein, and we made protein clusters. We then eliminated all protein clusters that contained proteins from an animal or a non-photosynthetic organism. So we were getting rid of all those proteins that had decent homology to animal or non-photosynthetic proteins. And what remained were clusters of proteins present in green lineage photosynthetic organisms, present in plants and green algae, but absent or highly diverged in non-photosynthetic organisms. We also pulled in paralogs to this, but this is called the green cut. And these are proteins really that have some specificity to green lineage organisms, and they may be also present in other photosynthetic organisms. We just didn't use those in the analysis. For example, they may be in red algae, they may be in cyanobacteria, they may be in diatoms. Okay, and this just shows you another way of thinking about it. The green cut proteins are present in these organisms, but absent in these organisms. So an easy visualization of what the green cut really is. And we could go further, and we can say that there are now 597 green lineage specific genes. And of that green cut collection of genes or proteins, about 50% have unknown functions. Now, many of them are called knowns, but they're also not really knowns. You could say something is a phosphatase, but you won't be able to say what it does in terms of biological function. So there are many in that category as well. If we break down the green cut into categories, knowns and unknowns, you could see here that for photosynthesis, we could identify 62 proteins that are clearly involved in photosynthesis in some way. We know the function of 58. We don't know the function of 4. And of course, photosynthesis has been heavily studied. If you look, however, at proteins involved in signaling in the green cut, you could see there are 16 proteins here that are unknowns with 9 knowns. So most of them are unknowns in terms of function. Anything to do really with regulation, like nucleic acid transactions, 39 that are unknown, 32 that are known, there are more unknowns than knowns. So we have a lot of unknown function in this green cut, and the suggestion that some of those, or many of those, may be associated with photosynthetic processes. Now, of course, while we can identify mutants defective for photosynthetic function, it's really complex to understand what those mutants do, because many processes could affect photosynthesis. You could inactivate proteins involved in electron transport, or even downstream involved in the export of reductant to mitochondria, or the dark reactions, the Calvin-Benson-Basham cycle, or carbon storage, such as starch and lipid synthesis. So you would get photosynthetic phenotypes for a lot of different mutant types. Now, we obtained, though, a lot of green cut mutants by both forward and reverse genetic screens. And with the new mutant library that Martin Jonikis has made, we have many of the majority of the green cut proteins as represented by mutant alleles. And one of the ones we were interested in for a while is a green cut protein called CGL71. Again, it's conserved in green lineage organisms. That's what the CGL stands for. Now, CGL71 is in the chloroplast. It's in the thylakoid membranes. It has a transmembrane domain, which goes across the thylakoids. And it also has TPR repeats in it. And these are tetratricopeptide repeats. And they're really protein-protein interaction domains. So this, you would conclude, maybe is a scaffolding protein. Maybe it's involved in putting something together. You could start to look at the phenotype of the mutant, the CGL71 mutant. It's inactivated right at the beginning of the gene in the first exon. And you could see wild type grown on minimal media, that is photoautotrophically, grows fine at different light intensities. The CGL71 mutant doesn't grow. So that suggests right away that this mutant is defective in photosynthesis. You could look at it in the presence of fixed carbon, and you could see wild type grows fine in the presence of fixed carbon. The mutant grows okay at low light, but at high light it dies. So it's high light sensitive. This also is typical of various photosynthetic mutants. So this suggests that this is a photosynthetic mutant. Now, you could do more analysis. This is a way to look at the efficiency of photosystem 2 at various light intensities. And here you could see the efficiency of photosystem 2 goes down markedly in the CGL71 mutant, and it stays higher in wild type cells and in the complemented CGL71 mutant. So we complemented the strain to show that the phenotype was actually caused by the mutation itself. So this suggests that PS2 is less efficient in this strain, but it's actually not a lesion in PS2. It's somewhere downstream from PS2 itself. And we know that based on the shape of this curve. If we analyze it more, we can go deeper into that phenotype, and you find that CGL71, this is an assay for photosystem 1. This is oxidation and reduction of photosystem 1. So in the light, you oxidize photosystem 1 in this assay. In the dark, it gets re-reduced. And you can see that CGL71, which is in red here, has much lower levels of active photosystem 1. So that lesion in CGL71 is downstream of PS2, and it looks like it's in photosystem 1. And then if you look more carefully at photosystem 1 and at the different polypeptides, so here are 3 polypeptides associated with photosystem 1. In the CGL71 mutant, you have considerably less of all those PS1 polypeptides, and you can get them back by genetically complementing it by introducing an ectopic copy of CGL71. So it looks like CGL71 is somehow involved in the assembly or stability of photosystem 1. So we found another protein involved in photosystem 1 assembly. And that's interesting in itself. But we did notice a couple of things. We noticed that the phenotype was a little bit unstable. That is, it fluctuated a little bit. We couldn't get exactly the same results all the time. And that was interesting because then we thought maybe it has to do with the different environments, the different cell densities. And we thought about the evolution of PS1 and what might affect PS1. And let's go to the evolution of photosynthesis a little bit on the planet. And if you look where photosynthesis occurred, actually oxygenic photosynthesis probably occurred about 2.3 billion years ago, somewhere really in here. The atmosphere then gradually became oxygenated. You actually filled, the oxygen was used to oxidize all the sinks, both in the aqueous environment and the terrestrial environment. And the oxygen levels gradually built up. And a billion years ago, you started to see a much sharper rise in the oxygen levels to ultimately about 20% oxygen. Now, if you think about photosynthetic function, oxygenic photosynthesis started here. There was no oxygen around. You were producing oxygen, but you didn't have oxygen in the atmosphere. And if you look at the photosynthetic apparatus, it's full of oxygen-sensitive cofactors. And what are those oxygen-sensitive cofactors? Iron sulfur proteins are oxygen-sensitive. And so at the time that you were building the photosystems, you were using oxygen-sensitive cofactors, and you didn't have to worry about oxygen. Now, when the atmosphere became oxygenated, all of a sudden you were in the presence of oxygen, and these cofactors are sensitive to oxygen. So over evolutionary time, you had to develop mechanisms to protect the assembly and the mature complexes from oxygen disruption. So that's the sort of hypothesis. And this doesn't only go for photosynthesis. This goes for any process in the cell that has developed over that time period.
Quick question.
Yeah.
What is the red versus the green line?
I think those are the upper and the lower limits. So some people have estimated this, some people have estimated this, and I think this represents the upper and the lower limit.
And they're both atmospheric, not the ocean concentrated. Okay.
Atmospheric, and you can see it's atmosphere. Right? Okay. So organisms had to develop strategies to deal with O2 and positive redox. Just like when you become an organelle, you have to develop strategies to deal with oxygen and reactive oxygen and redox. You also had to deal with it over evolutionary time. So CGL71 may be part of a machine that protects the complex from O2 during assembly. Is there any evidence for this? Well, this shows you PS1 activity in wild-type cells and in CGL71, kept in the dark under oxic conditions for 4 days. And you can see CGL71 levels go down a bit in the dark under oxic conditions, whereas the wild-type levels of PS1 stay the same. So PS1 is declining here, PS1 is staying the same in wild-type. Now, of course, we could complement this by putting in an ectopic copy of wild-type CGL71, and it stays the same. But we could also complement it chemically, in a sense. We could complement it by taking these cells and doing the same thing, but now making it hypoxic. And we make it hypoxic, CGL71, the PS1 in the CGL71 mutant comes back. And it comes back to about 70% of wild-type levels. So hypoxia rescues that CGL71 mutant to a good extent. You could see that at the protein level as well. For example, in the dark, under oxic conditions, here's wild type, here's your PS1 proteins, and here's CGL71. And you could see after 4 days in the dark, you're almost missing all of the PS1 subunits. And if you grow the cells, you put the cells under hypoxic conditions and do the same exact thing, you get back those subunits. And so that assembly process, those subunits, that stability of PS1 is dependent on the oxygen levels. If you reduce those oxygen levels, you get back the complex and activity. And so this just shows you how you basically have many cofactors involved in a holo PS1 complex. And some of those cofactors are oxygen sensitive, especially the 3 iron-sulfur clusters, which are present here and are involved in electron flow and the charge separation in PS1. So CGL71 is required, possibly, for proper iron-sulfur assembly under oxic conditions. So maybe there are other machines in the cell also that are required to maintain the stability of complexes or to assemble the complexes in an oxic environment. And maybe that has developed over evolutionary time. We're now doing screens for mutants that survive better under hypoxic conditions than under oxic conditions, thinking that we may get other factors like mutants in genes encoding other factors like CGL71. And there may be some information where some strains might be more amenable to growth under hypoxic conditions, but that's really in the process now. So conclusion from this part is some green cut unknowns are likely involved in assembly stability of the photosynthetic complexes. CGL71 may be involved in protecting assembly processes from oxic redox conditions, which may reflect the evolutionary history of photosynthetic organisms. We're now searching for other mutants in photosynthetic function that are less impacted under hypoxic conditions in the dark. Some may be involved in protection of assembly processes from oxidative disruption. Oxygen levels may have been critical in the evolution of a photosynthetic organelle, like a chloroplast or a chromatophore, and we talked about why oxygen is so critical to that integration, but also in the mechanisms associated with assembly of photosynthetic complexes. So the more general lesson that I learned as I worked at Carnegie is this lesson. And this lesson is not a lesson that's given to you by granting agencies or even colleagues, but But I think it's an important thing to mention that if we want things to stay as they are, that is, if we really want to stay on top of science, if we really want to do the best science that we can, things will have to change. We have to change. And, you know, I see that in myself, that by going into other projects, thinking in different ways, I feel much more able to bridge things, to integrate things. And I see that in some others who have stayed very narrow in focus and get frustrated and don't necessarily enjoy the science as much. So I think it's important to think, you know, to have a philosophy about science too. And this is something that Carnegie really gave to me, and it was expressed in this novel, The Leopard, by de Lampedusa, which is about revolution and change in Italy, in Sicily, actually. Okay, and now these are some of the acknowledgments. There have been many wonderful interactions I've had at Carnegie and at Stanford, including with Fritz Prinz, where we did a lot of the AFM work. He's in mechanical engineering. John Pringle, where we do a lot of work on corals. People in Paris, where we interact on photosynthetic analyses and the biophysics of photosynthesis. The people who did the work that I presented today, for the Paulinella work, it was Eva Novak and Ru Zhang. And also we were helped by people at Rutgers, Dana Price and Debashish Bhattacharya. And people who did a lot of the work on the green cut and CGL71 were Mark and Rick, but also Steve Herbert, Tyler, and Shai. And many others in the lab, of course, contributed to this as well. And we have long-term collaborations with people at Berkeley, like Chris Niogi. So I'd like to thank you for your attention, and I'd be glad to take any questions that you have. Thank you. There's one other thing I might say at the end of this. One other thing that I learned and I grew into as I do these projects is you begin to think about what's important in biology and what's missing. And one of the things I think that is very important, and maybe it's just being studied now, is biotic interactions. And, you know, we study organisms in isolation. A lot of the work we did here, it's with organisms that have been isolated, axenic. But we now know that these biotic interactions are critical for organisms to go through their life cycle. They're critical for organisms to get certain nutrients. And I think the next tier of biology, the next wave, will be toward trying to understand those type of biotic interactions. Okay. Now I'm glad to take any questions. Sharon?
I have a question back to the chromatophores. Another thing that the two organisms need to coordinate is their reproduction and division. And do you see, how does the chromatophore itself divide? And do you see any gene transfer of critical cell division proteins from the chromatophore genome into the nuclear genome?
Right, so we haven't looked at that, but I will say that a number of the genes involved in division are still on the chromatophore genome. I don't know if all of them are still there, but clearly it has to be coordinated, because as it divides, those chromatophores divide as well, and they go into the progenitor cells. So it is definitely coordinated in that way. And I think that's something that people will study more. You know, the other thing I would say about that system, while it's an incredibly interesting system, I think, it's slow. So it takes 5 days for that organism to double. Now, that's a hard system to work on. So when Eva was here, I pushed her very hard to develop another system. And there is another system now that she's using. It's not a photosynthetic system, but it's a bacterial system that is an organelle, and it's the trypanosomatids, which also have a bacterial-like organelle. So she's working more on that. So the question of cell division, and that has a 12-hour doubling time. The question of cell division will be better studied, I think, in a system like that. Yeah.
I'm really intrigued by the presence of chromatosomes in the cyanophores.
Chromatophores, yeah.
Sorry, the chromatophores. And I'm wondering if they operate CCMs, expect them to, given the low light intensity and their slow growth, but they have the 2 copies of at least some of those genes. I guess, do you know anything about that?
You know, they certainly have the carboxysome. We don't know if they have the CCM. I suspect they wouldn't need the CCM. They're growing so slowly, so they may have lost genes for the CCM as well. We haven't really looked at that carefully. You know, if they had the CCM, and you know, you might expect it to grow faster, although it might be other things that are limiting growth. But then you might expect them to lose some of the genes for that, maybe as well. John?
I'm fascinated about this gene transfer between the host and the chromatophore. I guess I wonder if you find any gene that has 2 copies of duplicated, one in the host and one still in the chromatophore.
Yeah.
You do have that?
Yeah, so I tried to give that on one slide where there are 2 genes. One is a carboxysome gene, and the other is a photosynthesis gene. And they're found on both the chromatophore genome and the host nuclear genome. So that's where I was saying maybe we're watching evolution in process, that in fact the cell is going to lose one of those genes. One is going to win out, right?
So following that, then the overall observation is more genes or most of the genes end up in the host, transferring, moving to the host rather than going backwards.
That's right.
What do you think is the evolutionary force?
The driving force, I guess, I must say, I don't understand the arguments that well, but the driving force is the loss of genes in the chromatophore without replacing them through recombination. And so it has to do with an argument of loss and recombination. Now, usually, I don't actually know how many copies of the genome there is in the chromatophore, but in a chloroplast, there may be 80 copies of the genome. So I would imagine it would be hard to lose a gene, but it's not. But this is one of the organisms, it's the DNA ratchet, or Murphy's ratchet, people call it. And maybe there are some people here in this audience who understand it better than me in terms of really being certain that that's really the reason that they tend to lose genes and get gene reduction on the organelle genome and retain genes on the nuclear genome.
Or is the host providing a better regulatory system that give
It might be, and
regulation?
it might be the regulation and the coordination. And it might be there's more optimal coordination through the host, which is maybe controlling a lot of what's going on. Yeah.
Yes. As you know, I know probably more about thyroidoxin than other protein families. There are members that originated, it seems, in the cyanobacterium and those that are in the host. The ones in the cyanobacterium didn't change that much.
Mm-hmm.
But the one from the host changed considerably. So the best example is thyroidoxin F, which was a nuclear origin. But it was modified to achieve, to carry out its new functions. Are there members like that that are transported into the plastic that one might say are lazy and didn't change so much? So they're really very much like the animal counterpart.
Okay. So I can't answer that question, but I could tell you one reason I can't answer that question. And that is, you know, when we looked at pre-sequences on proteins, we had to try to get genes from the host nuclear genome. And we did get some of them. But we started to sequence the host nuclear genome to ask questions like you're asking. And what we were told the host nuclear genome, based on what people had looked at, was about 80 megabases. So we sequenced it, and we did a pretty deep sequencing. And we were getting fragments of 500 base pairs put together. And then we did an estimation of the genome. Turns out the host nuclear genome is over 10 gigabases. It's a huge genome. And so really getting that whole genome will be a bit of a challenge. But we wanted to, and we tried. But at this point, I'm not sure who's going to invest in something like that. Jeff?
The Polyvanilla PSAE and PSAK, you show their transfer made in the cytoplasm. In a cyanobacterium, are they thylakoid lumen proteins? I think PSAE is on the inside.
PSAE is stromal. That's the docking site for, and K is stromal. Both are stromal, yeah.
So they wouldn't have had any targeting information whatsoever.
To go into the lumen.
In cyanobacterium.
Yeah, they wouldn't have had targeting information in a cyanobacteria, right.
See, Diane, do you have any questions?
Yeah, I have several. The first one is, I'm really curious if you're finding things in your symbiotic system that have clear parallels with other symbiotic systems, such as those that are in insects, where people are looking at, similar to as you articulate, kind of evolution, a snapshot of evolution. Are there ways that you can see parallels regarding, how it is when you have the separation of function between the host and the symbiont that's dealt with, that kind of cross-cut between these different
Systems.
examples? And what unique challenges do you see in your system that are going to give us special insights?
Okay, so I think, I'm sure there is overlap in certain gene loss that occurs, especially a transport function that's lost and other pathways that are lost.
Are there examples already in the literature that you can compare to what you're finding, where you see very clear parallels?
I'm sure there are. We haven't done it. But that is something that Eva wants to do, and also especially using the trypanosomatid system that she's working with, but also with the Paulinella system. I think, the difference that we have in a system like this is photosynthesis. And you're dealing with a process that's giving off oxygen, that's creating reactive oxygen, that's a So the big challenge that an endosymbiont host has in terms of relationship is, how do you maintain that relationship until you create the situation where you've done so well in integrating that metabolism that you get rid of the lethality associated with that interaction. I think that's what intrigues me about this system most.
Yeah, I can see that. What about with mitochondria? I mean, I realize it's going the opposite direction in terms of the redox chemistry, but nevertheless you get similar intermediates that do cellular damage, and that would also be a challenge there too, right? I
think that would be a challenge there, but I guess I don't see the mitochondrial energetics as quite as toxic in a way. I don't see the leaks quite as much as what happens in the photosynthetic system, where you have many points where light can trigger excitation, and that electron could go to oxygen and generate superoxides at pretty high levels. In photosystem 1, you could get pretty high levels of superoxide, or singlet oxygen in photosystem 2. And so I think you're right. You're absolutely right that you will get toxic intermediates in mitochondria. But I don't think it's the same level. And I think the control in the system like Paulinella is much easier in the sense that it's light. You could turn off the light, turn on the light, modulate the intensity of the light. So you could really manipulate that very precisely, I think.
What is the next step here? I have a bunch of other questions, but I'm not sure if now is the most appropriate time for me to ask. Broader in scope than about the details of your presentation.
Sure, yeah.
But I could answer the one question, what I think is the next step, one next step. And I think that one of the next steps is fairly easy, and that's to do proteomics on the chromatophore. Because we don't know what for example, we don't know what the nitrogen transporters are. Once we know what they are, we could identify where they came from, their phylogenies. We don't know a lot of the other proteins that are present in the chromatophore that are involved in control or that are involved in assembly. So all these things can be looked at if we actually do the proteomics, both of the soluble phase, the thylakoid membranes, and the envelope membrane. And I think that is the plan. I think Eva has actually started to do that. So that was the plan when she left, and she's really, I think, launched into that. So she probably has information on that already.
Okay, great. Diane, you'll get a chance to have more discussions in the next hour and a half with Arthur and the other members of the committee. So thank you, everybody. Thank you, Arthur, for your talk. Let's
Thanks,
give a
Diane.
round of